Hi, so in today's research log, we will be talking about a common problem faced in space, which is called space osteopenia. So let's jump right into this. So, space osteopenia, what is it? Space osteopenia is sort of like the osteoporosis for space. So, due to a limited amount of physical stress, because it's a microgravity environment, there isn't as much gravity acting on the astronauts. Due to a limited amount of that stress, there isn't as much of an opportunity for the astronauts to lose any bone mass. So, more specifically, bone formation and resorption are how the bone gains and loses calcium or mass in general. So, bone resorption is when it loses mass, and bone formation is when it gains mass. So, obviously, here there is much more resorption, but not as much formation. And it's not that there isn't any formation, it's just that the formation stays about the same, but the resorption very much increases. Or, in some cases, the formation goes down because they don't get enough exercise either, and the resorption continues to increase. So, in order to evaluate solutions for this problem, we're going to have to start with the calcium metabolism cycle. So what you have is the surface of a bone, and right now it's quite skinny, let's say. And so these are very much over exaggerated versions of cells, but let's say these are your osteoblast cells. All right? So what these have in them is tons of calcium. So then after a bit, they release their calcium and they're not as bulgy anymore. Now they're flat because they've released their calcium into the bone. That's why this huge lump formed, right? So then, in order to balance this out, a protein interacts with, or a hormone interacts with the osteoblast, which releases a protein. This protein causes the creation of osteoclasts. Now, what do the osteoclasts do? The osteoclasts attach to the bone, and what they do is they proceed with the process of bone resorption, and that's why this lump disappeared, because the osteoclasts attach. Here are the osteoblasts to the side. The osteoclasts attached and took away the calcium, and now they will leave. So in the case of space osteopenia, less osteoblasts are produced while more osteoclasts are produced. So now let's talk about solutions to this. And one obvious solution is intercepting this protein that causes the creation of osteoclasts. But there is actually a problem with that. So within the bone, there are these cells called osteocytes. And what osteocytes do is they tell the osteoclasts where they shouldn't proceed with bone resorption, because these are the weaker points in the bone. And if the osteoclasts come and start proceeding with bone resorption in the places of weakness in the bone, then the bone will start to crack. So that's why osteocytes are necessary. So if no osteocytes are produced, because osteocytes are essentially produced from osteoclasts and osteoblasts too, if no osteocytes are even produced and an osteoclast proceeds with bone resorption in that place, then the bone will start to crack and diminish. So we do not want that. So a common solution to this problem are bisphosphonates. What are bisphosphonates? So bisphosphonates, what's important to know about them is that they have two phosphate groups.
And the oxygens on the ends here have negative charges. These can be linked by a multitude of atoms, sometimes carbon. But what's important to know here is that these oxygens on the end have negative charges, and these negative charges give them a high affinity for bones. And so, when we're talking about so specifically, what I mean with a high affinity of bones is that they inhibit osteoclasts. And that may seem like a good thing because then bone formation works even faster, or not faster, but yields a higher amount. So, because these osteoclasts aren't producing as much, the osteoclasts, just like the osteoblasts, make osteoclasts when a protein is released from them after a hormone strikes them. Osteoblasts are also created from osteoclasts in the same way. So, since osteoclasts are created from osteoblasts, let's say this represents osteoclasts, this represents osteoblasts. Alright? So, this is how much we start with, and this is how much we end up with after 2 weeks during production. And then with osteoblasts, this is how much we start with. This is how much we may get to, but after another 2 weeks, these are also going to go down because there aren't as many osteoclasts. And now the solution may be more calcium, but when do we know to take more calcium in space to counter the effects of the reduction of the osteoclast from the bisphosphonates? So one way to know when the osteoclasts are working rather than the osteoblasts, and probably the most effective and the most recent method, is to use a method of sorting through isotopes. And so what I mean is there are 6 calcium isotopes, and if you remember this from your chemistry class, heavier isotopes, or isotopes with a bigger number, just mean there are more neutrons in the nucleus. So what do we know about heavier things? It's harder for them to diffuse. It's harder for them to precipitate. And so, how does this affect what we're working with? So, since it is harder for the heavier isotopes to diffuse and to precipitate and so on, when osteoblasts are working through releasing the calcium into the bone, what they end up doing is taking away all the lighter, softer tissues, which means all of the lighter isotopes vanish. And so this can be tracked from measuring the amount of the isotopes that I just erased from urinary amounts of calcium isotopes. And so then later, when the osteoclasts are working, they release the lighter calcium isotopes back in to where they were going. How I said precipitation first with the osteoblasts, this is diffusion working. So the lighter isotopes diffuse back into the bone. And so when the lighter isotopes are present, then we know that the osteoclasts work. And again, we can measure the osteoclasts are at work. And again, we can measure this by measuring the amount of all of these isotopes in our urine. So now we know when we need to take in more calcium and when we need to use bisphosphonates when we're talking about gaining enough calcium to keep our bones strong in space. But there's a pretty big problem right now in space. So take in a lot of sodium chloride. sodium chloride, and this is just salt, table salt. So we take in a lot of this in space. There's a lot of this in the food that astronauts eat in space. And the problem with this is that it inhibits calcium production. And so this is also one much simpler solution to the problem. Rather than taking extra calcium and bisphosphonates, you could just reduce your sodium chloride intake in space. Just don't put as much salt in your food. So that concludes this video, and thank you for watching.